we will move on to metabolism. So for metabolism, we are going to have to focus on big pictures. There's no way we can get into all of the small details. So anabolism is synthesizing molecules. So amino plus amino acid equals a dipeptide. You put enough of those together, you get a polypeptide. So anabolism is building larger molecules from small ones. Catabolism is what we're going to be focusing on today. It is going to be breaking down the hydrolyzing larger structures. And we started catabolism in the last chapter, where we were breaking down food molecules through digestion, and then we needed to break them down for absorption. Today, we're going to be looking at a different aspect of catabolism, which is cellular respiration. So this is where we're breaking down food in order to form ATP. Energy can also be stored in glycogen and fats, which can be broken down later. So this is where there are three stages in processing nutrients. These are what we were doing in the studying in the last chapter, at breaking proteins down to the level of amino acids so that they could be absorbed and used for further processing. So then this is what we're going to be looking at today, stage 2 and stage 3. So stage 2, we're going to be looking at the process of glycolysis. Sorry about that. So glycolysis is effectively breaking glucose into 2. So we're going to break a 6-carbon glucose into 2 3-carbon pyruvic acids. In the process, we'll form 2 ATP. Then in mitochondria, we have 2 processes. We're going to have the Krebs cycle, also known as citric acid cycle, and oxidative phosphorylation. All of metabolic reactions, or most, I shouldn't say all, most metabolic reactions are what are called redox reactions. So in a redox reaction, they're always paired. There is a substrate that is getting oxidized. And when we say oxidized, they are losing hydrogen atoms. So I am going to put atoms here because this is including both the proton in the nucleus as well as the electron that is orbiting it. We've spent so much time in acid base talking about hydrogen ions that right now I need you to also to change and shift your focus that we are back to hydrogen atoms. Okay. So we're going to be, when we talk about redox reactions, we're going to be focusing on hydrogen, not on oxygen. This has to do with electronegativity. So we're just not going to go there. And our reactions involve hydrogen atoms. Okay, so the oxidized substance loses hydrogen atoms because it's losing the electron that is part of that hydrogen atom, and it's losing energy. The reduced substance gains electrons, it's gaining hydrogen atoms and energy. We use enzymes, and enzymes catalyze these reactions, but they can't carry the hydrogen atoms. Okay, so they can make the reaction happen, but they are not the hydrogen acceptor or donor. Instead, we use coenzymes. These are derived from B vitamins, and these are the ones that will actually handle the hydrogen atoms, okay? So this is their oxidized state. Okay. And then they are going to be reduced and form, this one forms what's called NADH plus H, okay? And sometimes it's just written as NADH because it's simpler, but there's always going to be 2 hydrogen atoms involved. For FAD, we have FADH2, and both of these are the reduced form. So you do need to be able to identify the oxidized form, which is NAD plus or FAD, as well as the reduced form. So reduction means that they have gained hydrogens, they have gained energy. So these 2 forms are your high energy. Forms. So in a lot of these reactions, they are redox reactions where NAD is converted into NADH plus H or FAD to FADH2. In doing so, a lot of the energy in that glucose, which becomes pyruvate, which then gets part of the TCA cycle, but down the road, a lot of that energy is now transferred into these reduced coenzymes. Okay. So with oxidized substances, they lose electrons, they lose hydrogen atoms, and they lose energy. The atoms, and that's associated with an increase in energy. There are 2 mechanisms that are used to make ATP. One is called substrate level phosphorylation. And substrate level phosphorylation is going to result in low ATP production. It's kind of like a crude mechanism to form ATP. So we can do it either in something in so substrate level phosphorylation is you're basically breaking something into, you'll get some energy released and you'll make some ATP. Oxidative phosphorylation is going to be where we capture some energy in electrons, and it is going to go down the cascade. Substrate level phosphorylation occurs in glycolysis in the cytosol and in Krebs cycle in the mitochondria. The second way of producing ATP is much more elegant. This is oxidative phosphorylation, which occurs in 2 processes. The one that most people have heard of is the electron transport chain. That's really so we often refer to the electron transport chain as oxidative phosphorylation, but that's actually just the first phase. And then the second phase is using a hydrogen gradient ion, this time it is ion gradient, to produce ATP. The substrate level phosphorylation involves breaking apart substrates. Oxidative phosphorylation is the capturing of the energy in electrons. So this is, again, looking at this graph shows substrate level phosphorylation, where we'll have glucose converted to pyruvic acid. So essentially breaking glucose from 6 carbons into 3 carbons will provide us ATP. The citric acid cycle will also provide ATP, 1 per turn. It's pretty slow. And then we'll get over 30 ATP generated through oxidative phosphorylation, which involves the electron transport chain and something called ATP synthase. Okay, so 2 levels: substrate level phosphorylation, oxidative phosphorylation, both generate ATP. This one, substrate level, will produce low levels of ATP. Oxidative phosphorylation will produce high levels of ATP. Substrate level basically breaks substrates in half. Oxidative is going to harness the So now we're going to look at carbohydrates specifically, fats specifically, and amino acids. And really the heart of metabolism is going to focus on carbohydrate metabolism, because fatty acids and amino acids are going to feed into the Krebs cycle. So if you learn it with carbohydrate metabolism, you already know the majority for lipids and amino acids. So we spend quite a bit more time teaching carbohydrate metabolism, because as I said, once you learn it, the other two kind of feed into the Krebs cycle. Okay. So when glucose, so we broke the disaccharides into monosaccharides, transported them through the epithelium, and then galactose is going to get converted into glucose. So we mainly talk about glucose metabolism. Fructose also gets converted, although it's a slightly more complicated way. But essentially, everything ends up as glucose. So we focus on glucose metabolism. When glucose enters the cell, it gets phosphorylated. So this is an active process that glucose gets converted to glucose 6-phosphate. This effectively traps it in the cell and keeps the glucose gradient going. Okay. Can you, I'm sorry, I'm going to have to pause for just a second. Can you we're going to take okay, we're back. Okay, so glucose enters, it gets phosphorylated with an active process and trapped inside that keeps glucose concentration gradient going down and into the cell. So you may have, depending on when you downloaded this, there was a typo here. It said 38, it should say 32. So the oxidation, so complete oxidation of glucose is you take a glucose molecule at C6H12O6. You will oxidize it with, use oxygen in the process to end up with final products of 6 water, 6 CO2, and 32 ATP. And about 2/3 is going to be lost as heat. And that's actually pretty efficient. Okay, so seems like a lot, but it's actually a relatively efficient process. Complete glucose catabolism requires 3 pathways: glycolysis, which takes place in the cytosol; Krebs cycle, which takes place in the mitochondria; and then the electron transport chain and oxidative phosphorylation, which also takes place in the mitochondria. I've separated these two because, just as a reminder, glycolysis and Krebs cycle are substrate level pathways for ATP production. So last year, we really kind of did it by location. Oh, so glycolysis is in the cytosol, and Krebs cycle and electron transport chain are in the mitochondria. But in reality, these two, glycolysis and Krebs cycle, almost have more in common than electron transport chain. Okay, so we're going to go through each of these three pathways, and they have to keep kind of go in this pathway. Glycolysis has to start first, and then Krebs cycle, and then the electron transport chain. So glycolysis is anaerobic, meaning that it does not require oxygen to occur. So oxygen can be present, but it is not needed. So glycolysis is going to occur independent of oxygen, does not utilize oxygen. It occurs in the cytosol, and glucose is broken into pyruvic acid molecules. So these are 3 carbon molecules. There's there's 3 phases. The first one is sugar activation. So before we even make glucose, we have to put energy in. Okay, so sugar activation occurs because glucose, 6-phosphate, is phosphorylated twice. And each one of those is going to require an ATP. So we go from glucose 6-phosphate to fructose 6-bisphosphate, which is a biphosphate, and it's phosphorylated again to fructose 1,6-biphosphate. Now, you do not need to know all of these intermediate substrates. What you do need to know is the first step of glycolysis. We have to invest 2 ATP. Okay, so on our ATP count, we're at -2 right now. This provides the activation energy needed to start the pathway. So we're going to start with glucose. We have to invest or lose 2 ATP in order to start the process. Once we get to fructose 1,6-bisphosphate, then it is going to get cleaved into 2 3-carbon fragments. So you need to know that we invest 2 ATP, and then that gives us enough energy to break that 6-carbon into 2 3-carbon fragments. It can be one of 2 isomers, and these convert back and forth. My favorite is glyceraldehyde 3-phosphate. I think it's cool. And so, but it really doesn't matter. So we invest 2 ATP. That gives us the energy to break into 2 3-carbon fragments. Those 2 3-carbon fragments are then going to get oxidized and generate ATP. So we have an enzyme, and then it is going to convert either of these 2 3-carbons substrates into pyruvic acid. When it does that, it generates 4 ATP, and it is going to reduce 2 NAD to NADH+. After this, we now have 2 pyruvic acid, and then we have these 2 reduced coenzymes. So the final products of glycolysis are 2 pyruvic acids, 2 reduced NADs, so we have 2 of these, and a net gain of 2 ATP, because we produced 4, converting glyceraldehyde 3-phosphate and its other isomer into pyruvate, but we invested 2, so it's a net gain of 2 ATP. So what are the take-home messages, right? Okay. So the take-home for glycolysis is that we start with glucose. And glucose is a 6-carbon molecule. We have to invest 2 ATP in order to make 3 carbon fragments. When those 3 carbon fragments are converted and we produce 2 NADH plus H's for a net gain of 2 ATP. So know what you start with. You start with glucose. You end with pyruvic acid or pyruvate, either term is fine. And then you also end up with a net of 2 ATP because you produced 4, but you needed 2 for activation energy. And then you also ended up with 2 reduced coenzymes. So far with glycolysis, we're at a net gain of 2 ATP. And if you remember, we need to get up to 32. That's the total ATP that can be produced from glucose. So the supply of the oxidized form of NAD+ is limited. If oxygen is present, then NADH plus H can enter the Krebs cycle. It hands off its hydrogens and gets reconverted back to NAD plus again. If no oxygen is present, it still needs to get back to its oxidized form. But if there isn't oxygen present, the Krebs cycle isn't turning, and so it can't go there. Instead, it has to give the hydrogen atoms back to pyruvic acid, reducing it to lactic acid, which then diffuses back out. However, lactic acid, when oxygen is present, can go back to pyruvic acid, and pyruvic acid goes back to the citric acid cycle. So it's not ever really lost. It's just temporarily on hold. So we are going to continue as if oxygen is present. So we've made 2 ATP, and we have these 2 NADH plus H's kind of hanging out, ready to go to electron transport chain. And then we have 2 molecules of pyruvic acid. So we've got the products from glycolysis, and now they need to move on. So we're going to move on to the citric acid cycle, to Krebs cycle. So Krebs cycle occurs in the mitochondria. Its status as far as aerobic, anaerobic is kind of a mix. It will not proceed in the absence of oxygen because it's linked to the electron transport chain. But it in of itself does not utilize oxygen. So we still consider it aerobic metabolism because it will not proceed in the absence of oxygen. Okay, so we have pyruvate, which is the same thing as pyruvic acid. 2 names, we're getting used to that, right? And it is 3 carbons. It needs to get transported by this transport protein into the mitochondria so it can enter Krebs cycle. The first thing that happens is that it loses, it's decarboxylated. It loses a CO2. And the reason why is because Krebs cycle works on even numbers. You have a 4-carbon unit and it adds to a 2-carbon unit, and eventually you'll lose 2 carbons, carbon dioxide. But you have to always add in is a 2-carbon fragment. So pyruvate being a 3-carbon fragment is not going to work. So it loses the CO2 to get down to 2 carbons. Once it is at 2 carbons, it is going to get oxidized. It's going to go through a reaction and produce an NADH plus H, and it's going to combine with coenzyme A to form acetyl-CoA. So pyruvate is 3 carbons. It needs to get down to 2 carbons. Once it's down to 2 carbons, it joins with coenzyme A and becomes acetyl-CoA. Acetyl-CoA is going to be a molecule that is used in many, many pathways. So we have pyruvic acid, one of the final products from glycolysis, and we actually have 2 of them. Okay, so we're going to go through a cycle with 1 of them, and then we'll multiply those numbers by 2. So this is called the transitional phase because it's transitioning into the Krebs cycle. In the transitional phase, we produce a CO2 because we need to get down to 2 carbons. We produce an NADH plus H, and it gets transformed into acetyl-CoA to join with oxaloacetic acid, which is 4 carbons, to form citric acid. Citric acid is considered the starting point. Then we go through a series of reactions and we'll generate CO2, we'll generate NADH plus H, we will also generate an FADH2, and we'll generate 1 ATP. So pyruvate comes in at 3 carbons. It gets decarboxylated, so it loses a CO2 to get down to 2 carbons. It joins with the coenzyme A to become acetyl-CoA. This combines with oxaloacetic acid to then move into the citric acid cycle, where it goes through a series of reactions. In the process, we form 2 CO2 because we need to lose 2 carbons if we're going to pick up another acetyl-CoA. These are just going to be released and excreted in the air, as we exhale, expire. We also reduce or oxidize NAD to the reduced form of NADH plus H. We get 3 of those. We get 1 FADH2, and we generate 1 ATP. So then the next few slides, just restate it. We've gone from pyruvate, we've decarboxylated it, lost 1 carbon, it's now CO2, combines with coenzyme A to become acetyl-CoA. Acetyl-CoA combines with 4-carbon oxaloacetic acid to produce the 6-carbon citric acid, which is why this is also called the citric acid cycle. There are 8 remaining steps, so that's that circle and it has enzymes to catalyze each step. Intermediate products are referred to as keto acids. You do not need to know that we go from citric acid to isocitric to ketoglutarate. You don't need to know the names. This is just 8 reactions that occur that we can regenerate citric acid by combining oxaloacetic acid with acetyl-CoA. So we need to regenerate that oxaloacetic acid. We've made 3 NADH plus H, 1 FADH2, and 1 ATP. So from each pyruvate acetyl-CoA, so once we have acetyl-CoA, we get 2 CO2, 3 NADHs, 1 FADH2, and 1 ATP. Because all of this goes back to a single, what do we get from glucose? Glucose produces 2 pyruvic acids, so we double those, 4 CO2, 6 NADHs, 2 FADH2, and 2 ATP, acetic acid, and keep the cycle moving. We generate 4 reduced molecules. And why is that important? That's important because these are going to go off to the electron transport chain. And 1 ATP, which if you double that by 2, we're at 2 ATP. So for glycolysis, we got 2 ATP, a net of 2. Now for Krebs cycle, we also have 2 ATP from our original glucose molecule. So we're at a whopping 4. And again, we need to get up to 32. So how do we get from 4 to 32? And that is going to be the electron transport chain. It's going to be these. So now these are not very long. They're just a few minutes. So I do think they're actually worth playing. Oops, I gotta turn my sound on. Hold on.
Glucose continues in the Krebs cycle. Pyruvic acid molecules formed during glycolysis diffuse from the cytoplasmic fluid into the mitochondrion, but pyruvic acid itself does not enter the Krebs cycle. Pyruvic acid is oxidized, while NAD+ is steps, bonds break, and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by 3 molecules of NADH and 1 other molecule, FADH2. 1 step produces an ATP molecule. The 4-carbon molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the Krebs cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now primarily carried by the NADH molecules.
All right. So I do think that this is a good, it's like a minute long, and it goes through pretty well. How many ATP are produced, how many NADH, FADH2, this would be from 2 acetyl-CoA. And then per turn, you also get off 2 CO2, so this would be 4 CO2. Breakdown products of fats and proteins can also enter the citric acid. Talk about those when we get to fats and proteins. Also, cycle intermediates can be used as building materials for anabolic reactions rather than just catabolic reactions. So the citric acid cycle is this central oxidation, but also for other pathways to feed in and for it to also spit off to anabolic pathways. So it's just the central hub that keeps going. So with glycolysis, each glucose molecule generates 6 CO2, 2 FADH2, and 8 NADH plus H. And then this would actually be 4 ATP. Oh no, with glycolysis, sorry. And then we get to, then Krebs would add 2 more. We are going to be now focusing on, I'm not sure this one's correct, so I'll have to relook at this. calculation. So just ignore this calculation for now. Okay, but we do have FeDH2 and NADH plus H to use. So where are those used? Okay. So the electron transport chain in oxidative phosphorylation has two phases. Okay, so the first phase is to create a proton gradient using these high energy electrons in the hydrogen atoms. And then the second phase is going to use that gradient to synthesize ATP. The electron transport chain requires oxygen. This is the only pathway that actually directly uses oxygen. And this is because, so if you go way back and we, glucose ends up in the breakdown of to the end products of carbon dioxide and water. This is where we need that oxygen to produce water. So while this has to keep moving for Krebs to keep moving, and obviously if this part isn't moving, phase 2 isn't moving, it's the only one that directly uses oxygen atoms. The citric acid cycle will not run if oxygen is not present, but does not directly use oxygen. It is performed on the membranes of the mitochondria, and it uses reduced coenzymes from Krebs cycle as substrates. So in Krebs cycle alone, from each turn, we get 3 NADH plus H plus 1 FADH2, multiply that by 2 if you want it for glucose. These will then are going to deliver hydrogen atoms to the electron transport chain proteins. The first step, and I like actually, there's one more. Where is this? There we go. Okay. I like this diagram to help explain things. Okay. So you have 5 complexes in the mitochondria on these membranes. Most of them are going to be associated with metal-containing enzymes. But protein, so mitochondrial complexes 1 and 2 are going to accept hydrogen atoms from NADH plus H, split. So the hydrogen atoms, if you remember, have a proton and an electron. The complexes are for harness these electrons, harness these hydrogen ions, and then it is going to get split off to a carrier molecule. Okay, so first NADH plus H, FADH2 drop off their hydrogen atoms to complexes 1 and 2. Then the hydrogen atoms get split into a hydrogen ion and an electron. Then the electrons are going to get transferred from 3 to 4. And as they continue to get transferred, they lose a little bit of energy, and they use this energy to pump hydrogen ions into this intermembrane space. When they get to complex 4, they've lost a lot of energy and are now going to combine with with oxygen to make water. So the first step, those mitochondrial complexes 1 and 2 accept hydrogens from NADH plus H and FADH2. NAD and FAD can now return to glycolysis in the Krebs cycle. The second step hydrogen atoms are split into a hydrogen ion, which is a naked proton. All there is, there's no neutron, no electron. So just a proton and an electron. The electrons are passed down from complex to complex, and a little energy is released with each transfer. That released energy is used by mitochondrial complexes 1, 3, and 4. You don't need to know the numbers. To pump hydrogen out, creating a hydrogen ion concentration gradient. So this is the whole purpose of the electron transport chain, is to create a hydrogen ion concentration gradient. At complex 4, electron pairs combine with hydrogen and oxygen to form water. In this diagram, it's a little bit different, but kind of similar. We have the first step where we have NADH plus H and FADH2 donate their hydrogens to complex 1 and 2. Complex 1 is going to then split the hydrogen into a hydrogen ion, and then the electron gets split off and is going to continue to get passed through with these coenzymes. These are called, most of them are cytochromes associated with iron. And as each time they get passed, this is the amount of energy in that electron as it goes from one to another, to another, to another, to another. The amount of energy and as it gives off this energy, we're going to be pumping hydrogen ions and creating a gradient. Eventually we get to complex 4, and these low energy electrons are going to combine with hydrogen and oxygen to form water. So we have now established a proton gradient with potential energy. So we still haven't made any more ATP, but we're about to. Okay, so we went through this one. Okay, so electrons are split and carry along and end up as water. The hydrogen ions, which are now, so these are ions. Well, just like we do normally with the sodium potassium ATP pump, we are now going to use this as a hydrogen pump. And so the enzyme is ATP synthase, and it is going to use the potential energy stored in this gradient. So you've got this huge buildup of hydrogen ions, and those hydrogen ions desperately want to get to the other side. One, because it's a concentration gradient. Also, there is a charge gradient. They are just desperate to get across, but the membrane is impermeable to hydrogen ions. So when ATP synthase is open, which it usually is, these just flow through, driving this, what's called a motor, and generating ATP. So the proton gradient it creates a pH gradient and a voltage gradient. Basically, it makes this huge bank of hydrogen ions that is desperate to get across the membrane because of that gradient and the voltage difference as well. Hydrogen ions are attracted to the matrix side of the membrane, but the membrane is impermeable to hydrogen ions. They can only re-enter the matrix using should be a mitochondrial membrane protein. And that protein is called ATP synthase. And as hydrogen goes through, it drives that motor to phosphorylate ADP, ADP creating ATP. Okay. So protons, we have tons of proteins now. they are going desperate to get across that membrane, but it's impermeable, so they have to travel through ATP synthase. As they do, this turns and this turns. And then ADP is phosphorylated to make ATP. And the number of reduced, so NADs, H's plus H's, and FADH2 that enter the electron transport chain is going to result in the production of 28 ATP. This is another one of a quick video, which I think is worth the time.
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. NADH and FADH2 molecules give up their high-energy electrons in the third phase of cellular respiration, electron transport and ATP synthase, where most of the cell's ATP fuel is produced. The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy H toward oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of
Oh, sorry. Oh, no.
electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed-up hydrogen ions give up their energy when they diffuse through a special protein complex in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP.